Hello, Biol-16. All right, today we're going to work on the anatomy of the nervous system. I had to use a different format today, but I'm sure everything will work out just fine. So the nervous system allows our body to communicate basically through really rapid conduction signals, which are called action potentials. Ooh, this is going to be interesting. Okay, so action potentials. Oops, totally different here. Oh, I'm not sure I can write. All right, so the action potentials are the electrical signals that allow senses to tell your body kind of what's going on in the world. You can smell things, you can hear things, you can feel things. We'll be looking at that soon. Where does the nervous system come up from? We're just going to talk very briefly about the embryonic development. Why? We don't usually talk about that in this class. Well, it turns out that the brain is a unique structure in that it develops from a thin piece of skin that has a process called invagination that happens. This is where our brain comes from, folks. So you start with this piece of skin, which is a double layer of cuboidal tissue, which is made chock full of stem cells. And there are these other regions here, which have also kind of budded inwards. So imagine this being a round embryo with a few chunks in it. This neural fold turns into what we call the neural tube. And these little dots on the side will become what we call neural crust. And neural crust becomes some other special tissues in your body like melanocytes and so forth. So what we're looking at here by the second and third slide are what we call the ectoderm. The ectoderm is going to become lots of different parts of your body. This isn't actually the outside skin of your body only. Ectoderm actually becomes part of your ear, part of your eye. Sensory structures are derived from ectoderm. As you can see here, our brain is derived from ectoderm because ectoderm budded inwards, it invaginated inwards, closed up, and started making this neural tube, which becomes your brain and spinal column. Amazing, huh? So as time goes on, in this picture, what they've done is they've removed just that round section, and you can see the lateral view of what's going to be the human body. So this will become the head, okay, or the front of the head eventually. This will become, well, actually, all of this will be a part of the head, but this is the basic shape that the embryo is going to take, all right? So what the gray part's going to become is what we call the prosencephalon, or the forebrain. This is going to be your cerebral cortex that we'll talk about later, and the diencephalon. The diencephalon gives rise to the eyes, the midbrain, called the thalamus, the hypothalamus, the master structure in the brain that keeps you asleep and your different cycles, your circadian cycles organized. The mesencephalon, which is the midbrain. The midbrain is interesting because this is going to become kind of like the deepest parts of the brain. We'll talk about that later. And then the back part called the rhombencephalon, which breaks into the mesencephalon and the myelencephalon. Mesencephalon is the pons and the cerebellum. These two are so important for muscle memory. If you're a guitar player out there or any sort of musical instrument player, you have an amazing pons and cerebellum. If you were a bird, the pons really help the birds fly. It helps them organize their wing beat pattern. And then the myelencephalon, it turns into the medulla oblongata. So as you can see here, the eye cup is just at the junction of the forebrain and the thalamus. Amazing, huh? My degree is in neuroscience, so I kind of have a lot to say about this stuff. So what ends up happening is the neural axis in mammals are organized in an anterior, posterior, or rostral to caudal fashion. Rostral and caudal is more obvious in four-legged animals like a dog. Rostral is always front near the snout, and caudal is the base of the tail here. I mean, we'd say this is the base of the tail. This is the tip of the tail, sorry. So we have the same thing. It's just we walk around on two legs, and if you had me in class, you'd see me, yes, for real, walking around the desks on my legs and hands. But it's really important to understand that our nervous system and our skeletal system is actually organized really similarly among mammals. The nervous system follows exactly the same pattern. It just depends on whether or not you're two-legged or four-legged as to where your eyes are positioned, right? Obviously, your eyes will be positioned up higher than most dogs. And the organization of the spinal column might be extended depending on whether or not you have a tail. I mean, like, we don't really have a tail, but dogs, cats, simians, they all do. So the brain, the most complex organ in our body, it weighs about 3 pounds. That's, a lot for so much. It's a lot of complexity in a little bitty working space. It can be somewhat variable depending on your body size, but for most people, it's about 3 pounds. It has about 20 billion neurons, so that's more neurons than the stars that you can see when you're out in Yosemite at night. That's a lot of neurons. Each one of those neurons, and then of course the neuron itself, is what we call a functional unit of the nervous system. Each one talks to thousands, hundreds, if not thousands of other neurons. So if you think about it, it's like a really fancy processing chip in your brain. Our brain is so much better at processing than your computer. It's amazing. And the key to that processing power is how the interactions occur between neurons and other neurons and neurons in the cells of your body. So how do we protect your brain? Of course, the cranial bones, you guys studied those. Cerebral spinal fluid, the really important fluid that surrounds your brain and fills your brain. The cerebral spinal fluid is usually sterile. When it is not sterile, you end up with meningitis, which means that the cerebral spinal fluid is encased by the meninges. I kind of think about it like the Ziploc around your central nervous system. We'll get to that later. And then the blood-brain barrier separates the brain tissue from circulating proteins and potentially pathogens floating around in your blood, medications as well. So if you've ever taken, let's say, for example, Benadryl is a medication that can cross the blood-brain barrier and can make you sleepy. On the other hand, Allegra or Claritin, they don't make you sleepy because they don't cross the blood-brain barrier. And the reality is to downregulate your immune response when you're allergic to something, you don't need to tell your brain to do it. Your brain is not down-regulating the response. The medication's telling your nervous system, hey, chill out. It's pretty amazing. We'll get to that later. So let's look at the layers covering the brain. The meninges start with the dura mater. Dura mater actually means tough mother. The Greeks came up with that, guys, not me. Dura mater has 2 layers. The periosteal layer, which is lots of connective tissue, and that's what sticks to the cranial bones. When you do your sheep brain dissection, you may still have some cranial bones, bony tissue attached to the sheep brain. As you pull it off, you'll see that tight connection between the two. That's a big deal because this keeps the brain from like sloshing around in there. Let's hope no sloshing happens, okay? It's bad. And then you have the meningeal layer, which is also called the spinal dura. And then there's the subdural space. We'll look at a picture of this in a minute. There's the arachnoid mater, which is from superficial to deep second layer. The arachnoid mater appears like a spider web, and hence arachnoid. And you should see that when you do your dissection. And then we have the subarachnoid space, which is the space kind of within all those little spider web type layers. And then the pia mater is the thinnest layer, and this is the actual layer that adheres to the brain. It's like a thin skin encasing the brain. So will you be able to see this? Some people's sheep brains have had the dura mater trimmed away. When we order the sheep brains, we can't designate that. You will definitely see dura close to the spinal cord though, and it looks like a thick, it's almost like paper, it's so thick, all right? The arachnoid mater and pia mater you might be able to see as well, but the pia, don't be surprised if the pia, you can't find it. It's super thin, and you'll know that you've peeled off the pia if, number one, it looks like the blood vessels have been peeled away. Those stick around in the arachnoid mater, all right? The arachnoid mater has lots of blood vessels in it. The pia mater, you usually just don't see. The brain will look kind of shiny. You'll know that you've gotten rid of all the meninges if the brain looks shiny. So what's circulating around underneath the dura? Cerebral spinal fluid. It circulates through the brain and the spinal cord. Our body makes about 500 mils of it a day, and it's replaced every 8 hours. This is super important for cushioning the brain. It supports the brain internally, but it supports it a little bit externally, right? It's kind of a we call it a hydroskeleton, okay? Hydro for fluid, skeleton support system. It gives nutrients to the brain. It does pass around some chemical messengers like ATP and stuff like that, and it helps get rid of waste. When we sleep, that's the point in time where our brain actually gets rid of lots of the toxic byproducts that build up over the day. And the more you think and the more you use your brain and the more stress you have, the more toxic byproducts you have actually in your brain. And so by sleeping, your blood pressure goes down and certain channels open up, allowing the cerebral spinal fluid access to that garbage that you need to get rid of, and it helps wash that away. If you don't get rid of that garbage, it's been shown that people who don't sleep enough, let's put it that way, 7 hours a night. If you don't sleep 7 to 9 hours a night, you don't get rid of all that garbage, and that's not a good thing. It can lead to, it's linked to, not lead to, that hasn't been shown yet, but it is linked with the incidence of dementia in aging. So get your sleep, you guys. Cerebral spinal fluid is supposed to be colorless. What I've seen of it, it can be colorless or kind of have a slight amber tint to it because you have urea in it. Urea is what makes our urine yellow, right? There's proteins in there as well, so it kind of tints it a little bit. But if it's like yellow, that's infection. That's bad. So here's the cerebral spinal fluid and where it's produced. So here's a capillary. Capillary leaks out, capillaries in our body leak out ions, vitamins, water, okay? And that all enters what we call a layer of choroid plexus. Choroid plexus is a tissue that produces cerebral spinal fluid by filtering out what comes out of the plasma in our circulatory system. So cerebral spinal fluid is a modified plasma. One of the things that I hope you take away from this class is every fluid in your body comes from the water you drink. You drink the water, goes into your stomach, into your digestive tract, where it's absorbed into the bloodstream. It circulates around. It gets, you get ions, nutrients, other things pumped into the bloodstream, good, healthy stuff, right? The stuff that keeps you alive. And then that gets pumped into different parts of the body. It becomes your extracellular fluid. It becomes lymph. Here, it becomes cerebral spinal fluid. We're looking at the production of cerebral spinal fluid in the third ventricle of the brain. We'll look at the ventricular system in a little bit. And what you'll find is in the third ventricle, lots of sodium, potassium. Your brain uses that like crazy. Calcium, magnesium, mostly calcium. It uses calcium a lot too. Of course, vitamins, nutrients, oxygen, tons of oxygen. Your brain is majorly metabolically active. I would have put glucose in there because your brain's a sugar monster. It loves sweets. It's like a toddler. It's actually more like a teenager. Man, do my teenagers like sweets. Waste products, things like amyloid, depending on the size of the protein, can get redistributed into cerebral spinal fluid, extra amino acids, like aspartame and stuff, gets pumped back into the bloodstream for circulation and elimination through the urinary system. So if you look over here, you'll see a side view of a capillary, right? One of the cerebral capillaries, and it's surrounded by other sets of cells. What's going on here? Well, these two gray cells represent gray matter, and these two yellow cells represent types of glia called astrocytes. Now, you can see here between this cell and this cell, down here in the choroid plexus, a gap. That's how the fluid escapes capillaries, through these gaps. Man, do I wish my pen was working right now. Let me see if there's any other way I can get my pen to work. No, it doesn't want to work. Let's see. Nope. Okay. If you look at a side view, you can see these tight junctions here. The tight junctions keep proteins out. They keep large things out, but allow ions and smaller medications like that Benadryl to leak into the brain. In order to stop that, astrocytes use what we call little end feet. They plug the hole. They just plug up that hole so stuff doesn't leak through very easily, and that is the blood-brain barrier. So the circulation of cerebral spinal fluid is essential to cushion the brain. So here's the third part of the third ventricle over here, okay? Cerebral spinal fluid's made right here in this third ventricle space. It enters the superior sagittal sinus where it circulates around. So all the blue represents cerebral spinal fluid. There are other places where you'll see choroid plexus producing cerebral spinal fluid. Over here, it's at the lateral aperture, leaking into the space, the dural space surrounding the spinal cord. Look at how short that spinal cord is. Yeah, that might be my spinal cord. I'm not very tall. And so the third ventricle, excuse me, and the lateral aperture produce tons of cord plexus, but you'll find cord plexus in other areas of the brain distributed every now and again. Here are the arachnoid granulations. The arachnoid granulations are other points where cerebral spinal fluid can escape into the what we call the subdural. Here's the dura mater. This is the subdural space, the superior sagittal sinus. Okay, and so you see right here in the subarachnoid space. Here's the arachnoid mater. Right, looks like spider webs. It allows the fluid to leak out. So each one of these little granulations keeps the pressure, not blood pressure, but cerebral spinal fluid pressure appropriate to cushion your brain. So whenever you are, let's say, on your skateboard or on a bike and you have to stop real fast, remember the law of motion means that things inside stop after the rest of you stop. So as your skull stops, your brain is still moving forward. And that cerebral spinal fluid helps cushion that movement. Pretty cool, huh? So the cerebral spinal circulation through the third ventricle enters down here, circulates around the cerebellum, up through, you can have through the granulations, you have an escape or a leakage of cerebral spinal fluid, so it goes underneath the subdural space and so forth. Okay, so what is going on with the brain here? The blood supply to the brain is essential because the brain requires lots of oxygen and glucose to survive. There are 2 vertebral arteries, 2 internal carotid arteries, and just tons of capillaries throughout the brain. There are tight junctions among the endothelial cells holding the brain together. So, sorry guys, hold on. Now, the major vasculature inside the brain is called the circle of Willis. So the circle of Willis has a number of very large blood vessels, like the ophthalmic. Well, the ophthalmic artery branches off the internal carotid. The internal carotid is what you're going to definitely need to know for upcoming maybe quizzes and so forth. There's the anterior cerebral artery. So anterior, where are you going to see that, right? That's feeding the cerebral cortex, which is up front, okay? Anterior spinal artery, posterior inferior cerebellar artery. The nice thing about all these names is that they describe what they're not innervating, but what they are sending circulation to. So definitely at least know the large blood vessel names. That'll help you later on. Now, the sinuses, we looked at the subdural sinuses a little while ago when we were looking at the kind of the cerebral spinal fluid circulating around. When you look at the subdural sinuses, you'll see that the veins are associated with the actual sinuses. So here's the superior sagittal sinus right there, and then you have the circulating cerebral spinal fluid, associated with it. Here's your dura mater right there. Now here are the cerebral veins. Now remember, veins are taking blood back to the heart. So here we were looking at blood coming out to the brain. This is blood returning back to the heart. All right, so the blood in the drain, the blood in the brain drains into the jugular vein. Say that 9 times really fast, or even 3 times. The blood from the brain drains into the jugular vein. Yep, there you go. The meningeal layers are essential in a lot of the venous circulation because they're associated, that sinus is associated with some of these veins here. So you'll see how these veins intercalate down into the arachnoid trabeculae, which we didn't really spend any time looking at because we don't have forever, unfortunately, to do this fun section. But the veins of the brain, oh my gosh, we've got to move on to another section, are associated with the sinuses, the superior sinus. Okay, now the blood-brain barrier I mentioned before. The blood-brain barrier is the junction between astrocytes, which are a type of glial cell, and the endothelial cells that line the capillaries. Now capillaries, for the most part, are just a series of squamous endothelial cells sitting next to each other. So the astrocytes help maintain that blood-brain barrier, which we saw a few minutes ago. Sorry, I thought I had another picture here. They can also wrap around capillaries. They can send their fingers around capillaries, again, plugging up the gaps between the cells of the endothelium. Now, the blood-brain barrier is pretty much intact with the exception of the hypothalamus. You can't have the blood-brain barrier completely tight when hormones need to escape the hypothalamus to regulate the body. The pineal gland, which produces melatonin, the pineal gland needs to receive serotonin, which then gets converted to melatonin. And believe it or not, some of the serotonin that's made in our body is made in our gut, and it enters our circulation and goes to our brain. So the pineal gland also has a much looser blood-brain barrier, allowing hormones to release into the blood, but also to receive some as well. And then the choroid plexus itself. The choroid plexus, which we looked at, is a series of modified ependymal cells. They have tight junctions amongst each other, okay? So things should not pass back and forth through the choroid plexus. The choroid plexus has a totally different leakiness or non-leakiness mechanism than capillaries do in the brain. Okay, so now let's talk about the different components of the brain. So the cerebellum, the cerebrum, are the two biggest parts of the brain. The cerebrum are the cerebral hemispheres, and they do have like superior and deeper, not superior, superficial and deeper structures. The cerebellum is at the base of the brain, really important for motor function. Then the mesencephalon, pons, diencephalon, which includes the thalamus and the hypothalamus, and the medulla oblongata, are all a part of the brainstem. Now, the brainstem is, you'll see that it sticks out of the base of the brain, which we can look at right here. But the brainstem will extend down into the spinal cord, okay? So the brainstem, this region here, is the junction between the spinal cord and the brain itself. The cerebrum itself is the largest part of the brain. It is most obvious in terms of having all these little zigzags on them. We'll get to that in a little bit. So the cerebral cortex is the most superficial part of the brain. Now the corpus callosum we'll look at in a few minutes. That is the section of the brain that's basically a bunch of wires, a bunch of axons connecting the right hemisphere of the cerebrum to the left. And this kind of slice down the middle, this separation between the right and the left hemispheres, is called the longitudinal fissure. So now let's look at the lobes of the cortex. It's pretty easy. You guys have actually looked at or named some of these items before when you were doing the bones of the skull. So we have the frontal lobe, which is in the front. The postcentral gyrus, we'll get to in a few minutes because that's important. The central sulcus, which separates the frontal lobe from the parietal lobe. There's another section here called the postcentral gyrus. We'll talk a little bit about that. The parieto-occipital sulcus is a line that separates the parietal lobe from the occipital lobe. And then you have the temporal lobe here that is separated from the frontal parietal lobes via the lateral sulcus. So this is what the brain that you're going to be dissecting looks a lot like. This is a human brain, but the sheep brain, believe it or not, looks a lot like the human brain. The only difference is you're going to see very large olfactory bulbs sticking out the front, but we'll get to that later. So here's the longitudinal fissure separating the two right and left hemispheres. The cerebral cortex are all these little zigzag lines. The gyrus are the tops of these elevations. They're elevated ridges. Down here, these little like valleys or depressions are called sulci, or an individual is a sulcus. The central sulcus goes all the way down the lateral side of the brain. These other ones, they just kind of zigzag around. The gray matter, which you won't be able to tell quite yet, makes up this, or consists of all the cell bodies or the neurons inside the brain. And the white matter are the axons, the extensions leaving the cell body. Now, earlier on, I had mentioned to you that when you're peeling away the dura mater and the pia mater and arachnoid mater from your sheep brain, it's going to look like it has a lot of blood vessels. And that's what you're seeing here. The cerebral veins associated with the arteries, and arteries actually, but you might see a lot of, you won't be able to tell because they'll be full of old blood. You won't be able to tell if they're arteries or veins. They'll just look like blood vessels. And they're associated with the arachnoid mater. As you remove that, gently peel it away, it'll reveal a nice, shiny, beautiful brain underneath. All right, so now let's look at an artist's rendition of the 5 lobes of the brain, if I can get this to set up right here. So you have the frontal, parietal, temporal, and occipital lobes, okay? Where's the 5th one? It's called the insular cortex, or insular lobe. Insula for internal. So if you peel away the two different sides, the temporal lobe here, the edge of the temporal lobe. There's another folding underneath here that will reveal gustatory cortex. This is the insular lobe here. It's underneath all of this other good stuff. Now, what's interesting about that, being as it's gustatory cortex, or that's, taste, basically, it's in a deeper part of the brain, which suggests that evolutionarily, that's been very important. And I don't know about you guys, but taste is pretty important to me. So I like that my gustatory cortex is protected by frontal cortex. That's just me, though. Up front here, in the most, the very, very front of the frontal lobe is the prefrontal cortex. This is the part of our brain that kind of gives us our personality. It kind of helps us make decisions. It gives you that kind of joie de vivre, that who you are, okay? The prefrontal cortex is the last part of the brain to wire up and become completely mature. Some people, oh my gosh, I swear, I heard this comedian this weekend, and they talked about, and she was right, the prefrontal cortex is the part of the brain that develops the slowest and the last in adults, Okay, I was like, yeah, that's true. And she goes, it fuses the last. I'm like, it fuses? Good Lord, no. The right and the left prefrontal cortex are separate. So I almost, oh my gosh, I almost jumped out of my skin. That was just me, though. So over here, the frontal cortex and prefrontal cortex mostly are helping you make good decisions. This kind of orange region here is called the somatic motor association area. This is where you feel things. This would be like sensory experiences, pressure, touch, to some degree sight, and other things. We'll look at a homunculus in a little bit. Here is the primary motor cortex. This is the part of the brain that tells your muscles to move. This is your voluntary connection, the info out, telling your muscles, okay, time to move. The primary sensory cortex here, associated with the postcentral gyrus, this is also associated with things like, this is actually a lot more pressure, pain. This is info in about the world around you. This is info out, but this mostly is sensation. It's not vision. It's not sound, okay? This somatic motor association premotor area kind of helps you plan, I guess you'd say. It gets information about sensory environmental cues, temperature. Is it windy? And it helps you plan what you're going to do in terms of movement. The parietal lobe extends back here into an association area, the somatic sensory association area. This is the part of your brain that actually puts things together for you. So I'll tell you a quick little story. I had a concussion a couple of years ago, and I whacked my head on the left parietal lobe, okay? And I couldn't remember words anymore, and I couldn't speak well. I was actually, and speech is over here in the temporal lobe. I couldn't speak well, but it wasn't because I couldn't talk, because if someone asked me a question, I could answer it. But I had a hard time initiating speech, and I couldn't name things. So like I needed a tablecloth because we were camping, and you want to put a tablecloth on those tables out there at your campsites. And I couldn't think of the word, so it was crazy. Back here is our visual association area. This is the part of the brain that sees a dog and puts the word dog there. And then this is the occipital lobe, the actual visual cortex. Over here, we have the auditory association areas and auditory cortex, and then extending down into the lower part of the temporal lobe. Okay, so now let's open up the brain and cut it right down the middle, the midline. Be careful when you do this on your dissection, because it's very easy to kind of, with a scalpel or a pair of scissors, if you have a knife, like a steak knife, a serrated steak knife, some people use that. The only problem is that it leaves serrations on your corpus callosum that could be misleading when you're trying to identify items, because then it'll have texture that the corpus callosum doesn't. Anyway, so let's look at the brain as we cut it down the sagittal line. So here's your, oops, let me get my pen here. Here is the forebrain. Blast, right? Here's the forebrain, or the frontal lobe. Back here is the occipital lobe. Down here is the cerebellum. Now, in your sheep, the cerebellum is going to look larger. This is a human brain preparation. It's not that necessarily the human cerebellum is so much smaller. It's just that the human cerebrum is expanded. It's much bigger in humans and in primates than it is in sheep. So you're going to see less cortex, cerebral cortex in sheep, and proportionally a larger cerebellum. Within the cerebellum, you'll see that, I mean, frankly, I think this looks kind of like cauliflower. Don't let that freak you out. Here's the fourth ventricle that contains cerebral spinal fluid. It's connected to the third ventricle up here. The cerebellum itself has the arbor vitae, arbor meaning as in the arbors of trees. It looks like branches of a tree, right? And if you look at the exterior surface, you'll see much more of a lined organization versus the zigzag sulci and gyri organization. So I'm starting posteriorly here. Here's what we call the aqueduct of the midbrain. The aqueduct is a connector here that allows cerebral spinal fluid to enter down into the fourth ventricle. The superior colliculus, inferior colliculus, you would need to know that in upper division neuroanatomy. Here is the parieto-occipital sulcus, the line that separates the parietal lobe from the occipital lobe. So here's your parietal lobe. The central sulcus separates your motor cortex and somatosensory cortex. Here is our parietal lobe again. And then here's the frontal lobe and prefrontal cortex. What do you need to know for the quiz? Well, a lot of these items, pretty much. You need to know the medulla oblongata, which contains a lot of regulatory centers for autonomic activation. So when we say autonomic, those are the automatic things that your body does, like breathing, heart rate, blood pressure as well, to some degree, is regulated through the medulla oblongata, which then extends down, which you can't see it here, into the spinal cord. The pons, the pons and the cerebellum work together in motor memory. We're not going to go into great detail there. In birds, the pons are much bigger. Like if we were doing a comparative neuroanatomy class, this thing is huge because it helps the bird see three-dimensionally while it's flying. For us, we see the world three-dimensionally too, but the world's coming at us in a different context. We're not like flying around. Fish, the ponds are pretty big as well. Also, if you think about water, swimming in water and flying in air, water and air are both fluids. It's just air is thinner than water. Freaky, huh? Here's the mesencephalon, or the midbrain. Please do not ever call this the reptilian brain, because that's just an insult to reptiles, I think. The mesencephalon is very ancient, and yes, you do find less cortex in a lizard, let's say, than in humans, but the proportion of mesencephalon is larger. The mesencephalon is just there. It's just they have less cortex. What else do you need to look for? The mammillary body, optic chiasm, which you should be able to see on your sheep. The optic chiasm is going to look different from the olfactory bulb, which is going to stick out of the front. We don't have a large olfactory bulb in human. And in fact, some people confuse the optic chiasm with the olfactory bulb. Right here, all right, so the mammillary body. Here's the anterior commissure. The commissure is a connection between the right and left side of the brain. It's not as big as our corpus callosum here, which again, this is a fiber tract pathway. Fiber tracts are the same things as axon bundles. Okay, so basically these are the cables connecting the gray matter from the right side to the left side of the brain and vice versa. The intraventricular foramen is right over here. It's a nerve. It's a place for nerves to go through. Postcentral gyrus, central sulcus. Remember, sulcus is always a valley. A gyrus is always a peak, like the top of the hill. The fornix, cingulate gyrus, you don't need to know so much about that. Those are more higher level neuroanatomy terms. The thalamus you do need to know, because the thalamus is a part of the midbrain. The epithalamus, you don't need to worry about that. The hypothalamus and the pineal are very important, though. Hypothalamus, of course, regulating all kinds of bodily functions, goal-driven behaviors, sleep, hunger, thirst, definitely thirst. And the one thing that you are not seeing, that I am not seeing labeled here, is the pituitary. Sometimes in these preparations, in the brain preparations, and in your sheep, you might see a big lump hanging down here, down below the hypothalamus, which is right here. So the big lump is the pituitary gland. Sometimes it's sheared off when the brain is harvested from the sheep. Hopefully your pituitary is intact. If it is, when you are removing the dura and the remaining bone on the ventral side of the brain, be very careful. You're going to see this it looks kind of like a reddish lump, brownish reddish lump. That is the pituitary gland. So try not to shear that off. It's hard though. Okay, so what's going on in the cerebrum? So the functional areas of the cortex are the sensory areas that brings in information and does help us interpret it to some degree. However, the association areas, okay, like in the parietal lobe, help give you a memory to interpret that sensory information. So let's say you feel wind. You're first going to feel it. It's going to go up to sensory areas. Your sensory cortex is going to say, hey, I'm feeling something, basically. And then the association areas in the parietal lobe will go back and help you remember, oh, that's what wind feels like, okay, or water. That's what rain feels like. The motor areas are different because they send out the commands to your muscles to undergo voluntary movement. The motor areas do not innervate smooth muscle like in your gut. There is innervation and control from the medulla oblongata, but not from these motor areas. These motor areas generate and send motor commands to skeletal muscle. This is what gets us moving, okay? So pay attention to that as you are thinking about what is our brain telling our body to do. If it is autonomic, automatic, right? There are other more subtle regulatory mechanisms having your body take care of that, because you don't need to think about it. Motor areas, though, that's a situation where you're making choices, right? And again, there are association areas associated, obviously, with motor areas. This is the memory of what it is like to jump. So the association areas, prefrontal motor cortex, is going to say, okay, I want to jump now. This is the pattern that we use, motor area, to generate a jump. And this is about how much force we think you're going to need. And so there's a lot of planning that your brain is doing even before you initiate a movement. And I mean, if you could see me now, I tend to gesticulate a lot. I talk with my hands a lot. That is all motor planned. All right, my brain is planning to do that. Even though I'm not thinking about it too much, that's because those motor programs are already in there. Guitar players, string players, violin players, they have an expanded motor association area and motor cortex for their left hand because they've memorized where to put their fingers to perform sounds on an instrument. So the different chords, let's say. And there have been many studies that show that the right side of the brain, the right motor areas, are expanded in people who play string instruments if they're using their left hand. So it's an amazing phenomenon. So that's your association areas. And also personality comes from the association areas and judgment to some degree, but prefrontal cortex is a big part of that too. Now, symmetry or asymmetry in the cerebrum, right and left sides of the brain. It is true that the right and left sides of the brain have special wiring to accomplish special tasks, which kind of makes sense because it's a good idea to distribute the work, right? Speech, math skills tend to be more localized to the left. Broca's area, Wernicke's area, super important regions for speech. If something happens, if you get bumped on your left temporal lobe and there's like a problem there, you get a hemorrhage or a rupture, an aneurysm, you'll have problems maybe doing math, maybe producing speech. Now, yes, I gave you the example of my my parietal lobe bump, but that was association. Remember, I couldn't remember what the thing was called. I couldn't put a word to the item. This is where you actually just can't talk. You can't produce anything. Now, the right side of the brain, the right hemisphere, does seem to have a little bit more to do with music, artistic awareness. When we say artistic awareness, that's more like spatial skills, okay? Does this mean you're right or left-brained? Well, it's kind of an old-fashioned terminology. I don't particularly care for it, but people use it all the time. Let's just think of it this way. If you don't use both sides of your brain, you're not going to be able to talk, do math, study science, study English. You're not going to be able to understand music, much less produce it or draw a figure. So both sides of the brain are involved and they cooperate to assist in the planning and execution of complex activities. Having said that, you knock one piece out, you're going to have problems. All right, so the functions of the cerebrum in general. The frontal lobe contains that primary motor cortex, the parietal primary sensory, you can also call that somatosensory. And when we say primary, this is the primary out and the primary in. It's not the association areas. The occipital lobe, visual stimuli, temporal lobe, auditory and olfactory, and all the lobes have their own special association areas, which is a good thing because that means that you can localize planning, judgment of how to do things. I mean, it seems silly, planning. What do you mean? Primary motor cortex, that's just sending the shock to your muscles, just zap, right? But it's not somatosensory, the premotor cortex that helps you plan how much of a shock you need. Do you need to just touch your brother on the arm, or do you need to punch him in the arm? There's all kinds of stuff that goes on to plan that movement. I had a brother. We did a lot of planning. Yeah. Okay, that is the cerebrum. Now let's move down into the diencephalon. The diencephalon is considered kind of the middle of the brain, the core. It consists mostly of the thalamus and the hypothalamus. You can see here in this picture, we have our corpus callosum again, our fornix, which is down here. Don't worry about it. The septum, the anterior cerebral artery is down here. It's hard to see in these pictures, though. It's like right here. Okay, it's part of it. The artery here is actually devoid of many red blood cells, old blood cells, but you can kind of see it. The anterior commissure. We have the frontal lobe, of course, the optic chiasm, the crossover between the right and left eyes, the optic nerve. The tuberal area, don't worry about that. Infundibulum, which is important when you talk about certain goal-directed behaviors, don't worry about that. Mammillary body, which is important in nursing, things like that. Fourth ventricle, which connects through the aqueduct to the third ventricle. We have the cerebellum here, right? It still looks like cauliflower to me if you cut a head of cauliflower sagittally. Check it out. Go buy a cauliflower. Cut it sagittally. It looks like the cerebellum. Your hypothalamus, goal-directed behavior. And when I say goal-directed, these are all the behaviors that you need to survive, like drinking water, like searching for a mate, fatigue. When you are sick, your hypothalamus changes your temperature, right? So the hypothalamus does a whole lot of things. It gives you fever if you're sick, if you have an infection. The pineal gland, which is important in sleep. The thalamus, which surrounds the third ventricle. You can kind of see the third ventricle looks weird here. When you look inside your sheep brain, you'll see that this is a bit concave. The choroid plexus, you could actually make out right here underneath the corpus callosum. In your preparation, since it's been fixed for a long time in formaldehyde, it may not look feathery, but in a fresh brain, it looks kind of feathery. It's really pretty. And then you have the parietal lobe up top here. So what does the diencephalon do? Let's start with the thalamus. Basically, there's a right and a left side of the thalamus, surrounds the third ventricle, and this is an information relay station. What it does is it has lots of gray matter, so nerve cells, neurons, lots of white matter, lots of axons going to other parts of the brain. What it allows you to do is plan something and compare it to another activity that you've done before. Like, okay, I'm going to jump now. Let's make sure the jumping code is okay so that we're jumping. It's not a spell checker, but it's an action checker for what you're doing. And it checks that you're doing it right, and then boom, you can do it. So many motor actions go through the thalamus. So a great example of that is, and this is what I studied in graduate school, the production of birdsong. Why is that important? Baby birds learn to sing songs from their father for attracting females. And the better your song is, the more females you'll attract for breeding. And if you have a dad, that means he was successful, right? So evolutionarily, that means if you can copy your dad, odds are you can sing to girls too and get some mates, have babies, right? Big deal for the species. So what happens in the thalamus is they listen to their auditory cortex, somatosensory, motor cortex. They learn the song, but as they're learning, they start making chirping sounds themselves. And as they're chirping, they're basically sculpting their song to match their dad. Well, that sculpting, part of that sculpting is edited by the thalamus. If you make the wrong sound, that sound is going to go away. That note in the bird song goes away, and that has to do with the thalamus saying, no, 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 your dad didn't make that sound. Nix that, get this going. So the thalamus is really interesting. That's all I'm going to talk about with the thalamus. Now, the hypothalamus, much more involved in regulating, like I said, goal-driven behaviors, eating, drinking, sexual arousal. It is very important in pain. If you are suffering severe pain, your hypothalamus is basically going to be telling you, sit down. Also big in pleasure, a big region in regulating anger and actually rage. We'll talk a little bit about, maybe in a little bit, about other centers of the brain which are involved in fear, memory formation with negative input, which is important. I mean, you don't want to repeat negative experiences, right? So our brain has architecture that if you've been bitten by a snake before, when you see a snake, your brain's going to fire off a message saying, hey, you got bitten by a snake before and you got really sick. You may want to move away from that. Be afraid. So we'll get to the limbic system maybe in a little bit. Okay, so the hypothalamus has an inferior and is inferior to the thalamus. So that means it's underneath. Oops, let me get my pencil again. Hopefully you're seeing these little things. Here's the thalamus. So it's inferior to the thalamus. It contains the pituitary, which they're showing here. Again, it might be sheared off in your preparation. And then you have the hypothalamic regions here. These little lines indicate fiber tracts, which are axons between gray matter neurons in one region to another region. It connects very strongly to the endocrine system and the nervous system to integrate hormonal action and these goal-driven behaviors, the regulation of temperature, circadian rhythms when you get sleepy and so forth. And the hypothalamus and the pituitary make hormones that help us do what we need to do. So within the hypothalamus, you have the paraventricular nucleus up high, the preoptic areas. Preoptic areas are interesting because what we see comes through here first, and it actually tells our hypothalamus, hey, it's daylight, as the information is passing through to the occipital lobe. And so these preoptic areas are helping set our circadian rhythms. So as you're around in sunlight, those preoptic areas are getting that information saying, oh, it's daytime, no melatonin. Don't go to sleep now. But as sunlight diminishes at the end of the day and the light gets more dim, the preoptic area is not firing off as many action potentials, and that allows your hypothalamus to make, at that point, some melatonin. Well, the hypothalamus will talk to the pineal, so we'll just leave that there. The autonomic centers, which are parasympathetic, we're going to leave that alone for now. Suprachiasmatic, which is also important in regulating day length or measuring day length. And then you have the supraoptic. So the preoptic and suprachiasmatic all are part of setting your circadian rhythms. Tuberal, I always want to say tuberonal. It's tuberal nuclei, don't worry about that. Optic chiasm, remember, connecting the two sides. The right eye and the left eye to the brain, we'll talk about that. Well, we already talked about that, but there's your optic chiasm. The infundibulum is the stalk here, the little dangly part that connects the pituitary to the brain itself. So the infundibulum is just a little stalk. Now, the pituitary is broken into two components, the pars distalis and the pars intermedia. Some people call it the anterior and posterior. Pars distalis and pars intermedia are the old terms, but it's good to know them both. You never know what's going to show up on the NCLEX, right? And so what happens here is the pars distalis, I had to learn the old terminology, the pars distalis makes their own hormones and spits it out into the bloodstream. Super vascularized. This whole structure is super vascularized. The hormones that are secreted by the posterior pituitary, the pars intermedia, okay, well, the pars intermedia and posterior, they kind of mix around. We'll get to that later. The posterior pituitary receives hormones that are produced way up here in the hypothalamus. And so again, highly, highly vascularized. Lots of blood vessels here as well. But what's happening here is the neurons up in the hypothalamus are transporting the neurotransmitter or the hormone down into the posterior lobe and releasing into the bloodstream. So the anterior lobe makes the hormones. The cells in the lobe makes the hormone, puts them into the bloodstream. Here, the cells up here ship it down. Why? I don't know. I have not heard a convincing, well, I have heard a few ideas evolutionarily why they're separated that way. It may have to do with parts of the pars intermedia. It just, don't worry about that right now. That's evolution. Okay, now we're going to come to the mesencephalon or the midbrain, the part that some people call the reptilian brain. Please don't call it that. It's not reptilian. It's just the midbrain. Fish have it too, and fish aren't reptiles. There's my biology part of me coming out. Okay, so we're going to look at just a few parts of the midbrain, but parts that are super important. The corpora quadrinemia, I can never say that one right either. Quadrinemia, two superior colliculi, which are part of, again, measuring incoming information and coordinating visual reflexes. This is a part of what's coordinating your pupillary response, blinking to some degree. Your eyes actually move. It's called a saccade. Your eyes move around in your in the orbit just slightly. You don't even notice it sometimes, but they move around, and that's a visual reflex. So if a piece of dust comes into your eye may move around a little bit, and you might not even notice it. There's an inferior colliculus, which is auditory reflexes. What is an auditory reflex? You hear something and put your arm up. Auditory reflexes are a part of activating your visual reflex. If you hear something to your right, and like it's Walking Dead time and it's a zombie, your eyes are immediately going to shift right so you can see what that is. What's going on? Is there a predator there? Is there a zombie there? Oh, is there a kitty there, right? Is it your mom? Is it somebody bringing you some cake? How awesome would that be, right? So you have a coordination of visual reflex with auditory reflex in this case. The cerebral peduncles, peduncles are interesting because what they are motor tracks. What that means is that is a descending group of axons sending information down to your spinal cord, okay? So when you're looking at the peduncles, which are right here, I don't know if you can see my pen, superior cerebellar peduncle. The cerebellar peduncle is attached to the cerebellum, and that's kind of, again, having to do with routing motor memory information. The mid-cerebellar peduncle and the inferior cerebellar peduncles are here. So basically, these are all axon tracks, motor tracks. And then the superior cerebellar peduncles, they attach to the midbrain and the cerebellum. So we have both lateral and posterior views of the midbrain. Please call it midbrain. Please don't call it reptilian brain. I might even put that on the quiz just because I want to see how many people will remember that. So mesencephalon, what's going on with the gray matter? Remember again, gray matter are the neurons, the cells of the brain that do the work. The tectum helps integrate information regarding auditory and visual stimuli. So the tectum, we used to call them the optic tectum. See if I can pull it up back over here. Where's my tectum? It looks a little different. So I studied all of this in birds, and the optic tectum was huge. And they're not showing us, where's the tectum here? They're not showing us the tectum. Dang it. I don't want to go back too far. The walls and the floor of the mesencephalon have a lot to do with muscle tone, limb position, which means it's dealing with those receptors that are embedded inside of the tendons of the arms and the legs and your joints, basically. And it helps, that gray matter helps process incoming sensation, outgoing motor commands, helps us maintain consciousness. So when you're looking at the mesencephalon, part of what's going on, if you, let's say, are having surgery and they're giving you some sort of anesthesia, general anesthesia, this is part of what's going down, is your gray matter in the mesencephalon, the midbrain. If they can knock out your midbrain, you're going down. You're going to sleep, all right, which is a good thing if you're having surgery. White matter, again, these are all those axons, axon bundles. A way to think about an axon bundle is like a nerve inside of the brain. It's associated with the cranial nerves, actually, but axon bundles throughout the brain and fiber tracts are all basically the nerves of the brain. Now, the medulla oblongata is the connection between the brain and the spinal cord. All the information travels through the medulla, all the information coming and going, which means that's a pretty major pathway. You know, just sitting here and trying to remember everything to talk to you guys about with all this. I have all kinds of neurons firing off, but the information has to be processed and sent basically to my mouth, the rhythmicity center, so I can measure my breathing to speak. Sometimes I get excited, so my heart rate will go up. It's kind of funny. But anyways, so the physical connection, one of the connections goes through this thing called the reticular formation. The reticular formation basically is part of what keeps you awake, keeps levels of consciousness up and going. If you get sleep, if you're bored, the reticular formation sometimes in some individuals down regulates. It just kind of like isn't firing off as much. There are 3 reflex centers: the cardiac, the medullary rhythmicity center for breathing, and then the vasomotor center, which has a lot to do with your blood pressure. So like I said, the medulla is regulating to some degree your blood pressure. There is also local regulation depending where you are in the body, but your medulla oblongata is setting the basic tone of the blood vessel diameter. And it's connected through the pons, and here are our pons. Okay, so the pons connect the cerebellar hemispheres of the brain, midbrain, diencephalon. The pons basically help regulate the information going down and information going up. It's kind of hard not to get into too much detail because we're already at almost an hour here and I still have about 20 slides to go. So I'm trying to keep it light on some of these more superficial structures. So again, we have fibers, transverse fibers, which are fiber tracts, white matter. Here's our amniotic center and the nematoxic center. These are very, very important in regulating breathing. We have the reticular formation. We have the ovulatory nucleus, right? Because it's about the size of an olive. That's why they call it that. Here's the initiation of the medulla, cerebellum, fourth ventricle, and so forth. So lots of gray matter in the pons, regulating lots of respiration. So the pons, breathing. The pons are all about breathing. There is some in other animals, regions associated with vision. Don't worry about that with us right now. You're probably never going to need to know that, but I think it's fun. And there are 4 cranial nerves associated with the pons. All right, so you're going to need to know that for when you're studying your cranial nerves. The white matter, again, are your ascending and descending fiber tracts or nerves throughout the brain. Transverse fibers go between the 2 hemispheres. Now let's talk about the cerebellum. Like I had mentioned before, the cerebellum is really important in motor memory. Cerebellum has the right and the left side. The vermis separates the right from the left. The folia are the peaks, okay? We don't call these gyri or sulci. We just call these little things folds, hence folia. There's a posterior lobe and an anterior lobe. So you have to think about the cerebellum as upside down. It attaches, when you're looking at what we would think of as the top of the cerebellum, you're actually looking at it flipped up. So this is, the superficial surface is actually on the underside of the brain. We have the right hemisphere and the left hemisphere, so anterior, posterior, and there is a primary fissure that's really obvious. The fissures are obvious, I think, on the cerebellum. The arbor vitae are, again, it's the part that I keep telling you I think looks like cauliflower. I hope you still eat cauliflower after you're done with this. That's the white matter. Those are the axons, the nerves within the cerebellum connecting to the brain. The 3 peduncles, again, the peduncles are fiber tracts. They send information out through those peduncles or fiber tracts to innervate the midbrain, the pons, medulla, and so forth. And they help organize motor activities in the brain. It gives you a smoother movement. This is not a structure that's affected by Parkinson's disease primarily. Later on in the disease, in some patients I've read, it can be affected, but Parkinson's disease is a disease of the midbrain, which we're getting to now as we look through a frontal section of the brain. So the cerebral cortex have are more superficial, but deep in the brain, in the midbrain here, we have our basal nuclei. The basal nuclei consist of the globus pallidus, down low here. Don't worry about the red one right now. The putamen and the caudate nucleus. Here's the corpus callosum connecting the two. This is the lateral ventricle. This will be part of the fourth ventricle here, okay? And what are we looking at here? The caudate and the putamen make up together a structure called the striatum. The striatum is what dies in the Parkinson's disease, and we don't need to go into the details of it. But what it does is it's when people start demonstrating Parkinson's, some people, not all, they'll start their hands will start tremoring. Okay, there's a lot of reasons why tremoring happens, but this is one of them. Okay, as the disease progresses and gets worse and worse, the tremoring gets faster and faster, and eventually it actually stops. And it's not that the tremoring has stopped, that it's gone away, the disease is gone. What's happened is that the tremoring think of it this way, it's gone so fast that they're stiff. And that's what happens in Parkinson's. They end up being very, very stiff. It's very painful, I would imagine. Here's another side view, a lateral view of the hypothalamus showing, the diencephalon showing the thalamus, hypothalamus, and the pituitary, again, hanging down through the infundibulum there. This is in the human. It'll look a little different in your sheep. Here is the midbrain, pons, and medulla. So these are general maps just showing you where the midbrain and the hindbrain structures are. Cerebellum we've talked about already, beautiful structure. And again, in humans, it's kind of compressed down at the bottom, and in the sheep, it'll look bigger. And now let's talk about the cranial nerves. Do you need to know them? Yeah. Cranial nerve 1 is the olfactory nerve, which ends in the olfactory bulb. In humans, it's not really noticeable. Humans, we don't actually go around. I mean, even though I love the smell of coffee, and I'm drinking my coffee right now, the olfactory nerve is nerve number 1. In sheep, in fish, it's enormous because that's how they know where they are. Water has a smell, believe it or not. The optic nerve is nerve number 2. This is the nerve that will be associated with your eyes. This is not necessarily the optic chiasm, okay? The old oculomotor nerve is nerve 3. This is helping you steer your eyes to look at something. The trochlear nerve is nerve 4. The trigeminal nerve is nerve 5. The trigeminal innervates your face. You don't want to mess that up. The abductens nerve is nerve 6. The facial nerve also innervates your face. It's nerve 7. Trigeminal is more like it does a few things. I believe that's part of your jaw. Hypoglossal, vestibulocochlear, sorry, did I say abductens? The abductens is here, vestibular cochlear. That's balance and hearing. Then we have the hypoglossal, the glossopharyngeal, the vagus nerve, which I hate messing up because that one, gets me every time. Accessory nerve is the 11th nerve. Okay, so what do you want to know about each one of these? Well, you have an assignment identifying what information each nerve processes. Vagus nerve, this thing is brutal. It brings information regarding pain, things that happen in the periphery of your body, and the vagus nerve will shut you down. It'll make you pass out. It can activate a drop in blood pressure if there is severe pain. And the idea is if something really bad is happening and there's severe pain, let's drop your blood pressure so you'll lay down and chill out for a little bit, and hopefully you'll get better. At least that's my interpretation of the vagus nerve. All right, so now let's talk about the spinal cord. The spinal cord does have, we've talked about the cervical vertebra, the thoracic vertebra, the lumbar vertebra, and the sacral ending in the coccyx, okay? Basically the idea is, and now we've removed the vertebra from the spinal cord here. Now what happens here is the spinal cord goes down to the lumbar level, okay? But starting at L2, lumbar 2, the spinal cord gets thinner and thinner as nerves start emanating to innervate your legs and your lower abdominal regions, okay? So these lumbar nerves, as they fan out, are called the cauda equina, or cauda equina, if you want to think about it. It looks like a horsetail. That's what it means, horsetail. These nerves fan out to innervate different muscles and different structures in your pelvic region, in your abdominal region, and in your legs. Over here, up on top in the cervical region, we have something called the cervical enlargement. It's a really big kind of lump. Basically it's the nerves that are going to the neck and the arms. And of course, it makes sense that this is a little thicker here because we're doing fine motor skills. Our arms may be strong, mine not so much, right? But you might have a lot of muscle associated with your arms, and we do these fine motor movements with our hands. So the cervical enlargement references an area associated with your hands and arms, arms, neck. We have a lumbar enlargement, which is the lower pelvis, or from the pelvis to the lower extremities, and the regions in between innervate the thoracic regions in between the posterior median sulcus and so forth. Well, posterior median sulcus is the center, but the initial thoracic regions here are innervating respiratory muscles, lungs to some degree, maybe your liver and so forth. How many nerves are coming from each area? Well, remember we had the, how many cervical vertebrae? You eat breakfast at 7, lunch at noon, dinner at 5, right? 7, 12, 5. It's a little different here. When we're talking about the nerves, everything always occurs in pairs, okay? So the conus medullaris is a region where you're going to be seeing the tapered end of the spinal cord. That's the very bottom, before the cauda equina, okay? The filium terminale is the fibers that are going to extend from the conus medullaris, so just down, okay, from the coccyx. So keep going down. Here's the filium terminale down here. It's the very base of the spinal cord. It's just basically nerves. There are 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar. Oh, haven't we seen numbers kind of like this before? 5 sacral, 1 coccygeal. Sorry, I didn't roll over there. They all exit the vertebral columns through those intervertebral foramina. I always called them foramina, but I had a British anatomy teacher. And then again, the cauda equina is the tail, the horse tail, that sends nerves out beyond the conus medullaris. So looking at a cross-section of the spinal cord, which you guys started on last week, you can see that as you cut the spinal cord through, now remember, the spinal cord is different from the vertebra itself. This is the actual white and gray matter coming down from the brain, okay? There's a gray commissure, the central canal, which is filled with cerebral spinal fluid, the ventral median fissure, okay? So when you're looking at this, anterior and posterior, this would be closer to your belly. Posterior, this is on your back, okay, near your back. So here's the posterior median sulcus up here. The dorsal median sulcus, dorsal and posterior are pretty much the same thing. Now we have the dorsal and ventral gray horns. In some books, dorsal and ventral, they will call anterior and posterior. So over here, you see the posterior lateral gray horn, okay? We always called it dorsal when I was in school because we hadn't converted to the posterior and anterior anatomical terminology yet. But I was studying animals more. And in animals, they're on all four, like your rabbit, right? So on the rabbit, it's dorsal and ventral. In the human, it's posterior, anterior, because we're not down on all fours. It's our posterior, which is our dorsal, and anterior, which is our ventral, our belly, okay? The dorsal ventral rootlets, which you'll see heading out here. You can see a little bit of pia mater right there. Isn't that cute? So the pia mater extends all the way down the spinal cord to the conus medullaris. And then when we look at an artist rendition, it kind of looks a little cleaner, although this is such a beautiful butterfly shape. I always love looking at the raw sections of the spinal cord. Here's our dura mater, arachnoid mater, pia again. Beautiful, right? When we look at the artist rendition, you'll see the dorsal root up here. So isn't this funny? They're calling it the posterior gray horn, but the dorsal root, okay. It is the posterior horn. That sometimes it's called the posterior root. We have the somatic and the visceral sensory nuclei. Now, here's something interesting that you need to know. The term somatic always means muscle, skeletal muscle, okay? If it's somatic, it has to do with your sensory system, skeletal muscle, how your muscles feel, and to some degree also your epidermis. The visceral are your guts. Viscera are guts. So when you get punched in the gut, part of the pain you feel is somatic, right? Because there is external pain as well. But you also have tremendous visceral pain because that's the pain that's initiated as a consequence of a blunt force trauma to smooth muscle, to these really delicate structures in your abdominal region, right? I hate getting, well, I don't get punched in the gut very often, but, you know, my son and I do Taekwondo, and he sometimes kicks me in the gut, even with protection. I'm like, right? Quite something. So that's our sensory nuclei. The motor nuclei are all going out the ventral root or the anterior horn. Okay, so here's your anterior gray horn, hence it connects to the ventral root. Ventral, anterior, same thing. You have the somatic again. Look at all of that gray matter dedicated to controlling skeletal muscle. That's what that is. It's huge. Look at that. And then you have visceral. This is not necessarily controlling visceral smooth muscle, right, in blood vessels and in your digestive tract, but it's giving it an influence. We'll talk about that later when we get to the digestive system, and the circulatory system. Now we got to wrap this up, right? So looking at these cross sections of the spinal cord, it's really important to remember that the H shape or the butterfly shape is the gray matter, and this, all the white matter, is the stuff surrounding it. So in this particular preparation, they made the white matter purple, okay? That is how much information is traveling up and down your body through your spinal cord at any given time. And the butterfly is just the action part. Look at that. Isn't that unbelievable? That gets me every time. Nerve plexuses are really important in distributing the information from your spinal cord to your arms, your legs, and the viscera, guts, right? And one of the big nerve plexus, one of the huge ones, C1 through C5, is the cervical plexus. This is helping control your neck. The brachial plexus is a big deal as well because my son, for example, I can use him because he can't sue me. He's my son. He dislocated his shoulder last year. Since then, his shoulder keeps dislocating, and he keeps getting tingling and numbness in his hand. And it's because somewhere along his brachial plexus, it's pinching the nerve. And if that goes on for long enough, and it may be, and I forget which side, I think it's on the ulnar side. It may be pinching his ulnar nerve, but I can't tell because he's not really good at describing where the numbness is. He just kind of like wiggles his hand. Over time, those nerve pinches can damage sensory information and eventually motor information traveling down to your hand. Very problematic. So the brachial plexus is huge in controlling hand and hand coordination. We have the lumbar plexus, which is the pelvic area. The sacral plexus, don't think it just has to do with sitting down. This is where you're going to start seeing the sciatic nerve. We all have heard about sciatica, right? The fibular, the common fibular nerve, the tibial nerve, these nerves extend down to your calves, right? Femoral nerve, and so forth. So these nerves are controlling specific actions of the muscles in your femur. So the muscles associated with your femur, so your quads and your hamstring groups, the fibula and the tibia. Tibula. Man, I think I need more coffee. This lecture has just been crazy. Okay, so the vertebral column itself definitely surrounded by the meninges, the dura, the arachnoid, the pia mater. You'll see that in those slides. Spinal nerves structures are the dorsal root, bringing information in, the ventral root, sending information out, and mixed spinal nerves. Well, it's a mix of dorsal and ventral information to integrate what's going on. This is what I feel. Are we acting on it? The spinal nerves do have coverings, the epineurium, the perineurium, and the endoneurium. Epi is always on the outside. Peri is surrounding groups of axons, and endo surrounds an individual nerve cell. The endoneurium is made up of Schwann cells if myelin is involved, and most of them do have myelinated axons. So the spinal nerves themselves branch, and so the rami are the branches of spinal nerves. A ramus is a singular. The ventral ramus, or so the singular ventral, innervates skin, muscles, extremities. The dorsal ramus, dorsal skin, dorsal muscles. So you have separate ramies that are controlling the top of a structure and the bottom of the structure. And remember, dorsal and ventral is a little weird in humans because we have anterior, posterior, and these were defined originally in animals. Well, sort of in animals, also in humans. The rami have 4 networks or plexuses, and we just talked about them: the cervical, the brachial, the lumbar, and the sacral. Cervical plexus is C1 through C5, brachial plexus C5 through T1, lumbar plexus L1 through L4, and sacral plexus L4 through S4. And this will help you on your project, or not your project, but your lab assignment this week, which is identifying the function of the plexes. So if we look at a cadaver, look at those beautiful nerves. Holy cow. So the cervical plexus is up high. The clavicle was removed here. They cut through the clavicle so you could see the plexus. Here's the brachial plexus emanating out here. So when somebody dislocates their arm, we have the head of the humerus. You can't see the humerus too well here. The head of the humerus can pull on the plexus, and again, I'm not sure which part of the brachial plexus is being affected in my son right now. Boy, if I keep using this lecture, it'll be, 10 years from now, which hopefully it won't be for 10 years. Maybe I'll redo it. But the idea, wouldn't it be something that I'd remember? Oh, my son dislocated his arm. But we have a bunch of nerves traveling down to the radial nerve, the ulnar nerve, and the medial nerve. They're all connected up here through the brachial plexus. And here's an artist rendition demonstrating the lumbar plexus, the different names, the subcostal nerve. Do you need to memorize all these nerves? No, but you should know what they innervate to some degree, kind of the main areas. And then the sacral plexus down low. Now, what's interesting about these nerve plexes is that they innervate kind of a weird pattern of the body. If you look at the innervation of the spinal nerves on the body, it generates what we call a dermatome. So L1 innervates on the left side, on the right side, kind of the groin, part of the groin region, the hip, but it extends down here on the left side into the inner thigh, right? So it's kind of interesting to look at the distribution of innervation of nerves. Why does that happen? It's a huge developmental evolutionary thing that we don't have time to get into. But if you have time to look at dermatomes and how they develop, it's really fun. So what are the spinal nerves doing? They allow us to have reflex arts, which means that you have receptors out there in the periphery, like in your skin, that get information from the world. And if it's information that will damage your body, let's say you put your hand on something hot, you will immediately pull your hand away without even thinking about it, because there's a sensory relay in the spinal cord. The information comes into the spinal cord through the dorsal horn, okay, through the dorsal root, comes down, your spinal cord says, hey, that's hot. And it sends an immediate message to the ventral motor areas of the spinal cord saying, move your hand now. And it sends a signal up to the brain saying, hey, you touched something hot, so we helped you out here and moved the hand away. It's a quick way, a faster way of getting the body out of harm's way. So motor neurons convey information to muscles saying move, and that's the effector. The effector muscle was the thing, or the receptor is usually associated with the effector muscle. There are 3 types of reflexes: a stretch reflex, which is called monosynaptic. It's ipsilateral. Ipsi means same side, lateral means side. So ipsilateral means like a monosynaptic reflex on the same side of the body. Monosegmental, meaning it doesn't spread through dermatomes. Flexor reflexes are polysynaptic. In other words, there are many neurons involved here, not just like one neuron talking to another. There might be 5 neurons in this pathway. Again, it's still ipsilateral, same side of the body. Intersegmental, it can spread among dermatomes. The extensor reflexes, polysynaptic, contralateral, meaning it spreads to the other side too. Intersegmental, spreading through the dermatomes. Extensor reflexes will be things like walking. Flexor reflexes are picking things up with one arm. So here are the synapses. So a monosynaptic reflex, in the muscle here, there's let's say a receptor here that tells you something about the stretch of the muscle. It comes in, talks to the sensory ganglia, which talks to the motor neuron and causes the motor neuron to tell the muscle to contract. Here on the polysynaptic reflex, this is more like the sensory reception in your skin. It sends information to the sensory ganglia associated with the spinal cord. Two neurons are talked to talk to two muscle groups and say, hey, time to contract because we felt something. This is a much larger reaction. The polysynaptic reflex is a larger muscle activation than the monosynaptic reflex. Why do they exist? Polysynaptic basically gets you out of more trouble. Monosynaptic is coordination. The reflex arc that I just described to you, painful stimulus, instead of burning yourself, they gave you a tack, right? You poke your hand, it activates muscles in the or it activates neurons that extend up to the dorsal horn, comes back down here to activate muscle fibers in the effector. Boom, you're good to go. Helps you out there. So I know this was a really long lecture, and I'm sorry that it wasn't posted sooner. I hope that you're able to get through this in a reasonable amount of time, and I will catch you on the next chapter. Have a great day.